Okay. So good morning, everyone, and welcome to the Center for Theoretical and Computational Physics weekly seminar. Today, it is a pleasure to welcome Bruno Silva from the Iberian Nanotechnology Laboratory. Bruno is originally a PhD in chemistry from the University of Porto. This was followed by a postdoc at the University of California in Santa Barbara, where he worked with Cyrus Athenia on a variety of biophysics topics. And he then came back to Portugal and became a researcher at the Iberian Nanotechnology Laboratory, where he heads a research group. And he has very kindly agreed to tell us about his most recent work today. Before I hand over to Bruno for the truly interesting stuff, just a couple of announcements from me. First, this seminar is being recorded, so by staying on, you're giving consent to your voice and image being recorded and stored. Second, in the interests of internet stability, can I ask all of you, except the speaker, of course, to mute their microphones and turn off their cameras during the seminar. If you wish to ask a question at any point, you're of course free to unmute yourself, and if you so desire, turn on your camera as well. So now it's over to Bruno for the seminar.
Okay, thank you so much for the introduction. It is a pleasure to have the opportunity to present to you the work we've been developing here at INL. So today I will be talking, talking about three different topics related with lipid and polymer-based nanoparticles for gene delivery, which is our, let's say, main line of research. So these are very interesting self-assembled systems. So very interesting from a, let's say, fundamental point of view, but also quite relevant for society nowadays, as we have seen now with the outcome of this COVID-19 mRNA vaccines. So they are loosely based on similar systems that I'm going to speak here. So our group, we have a more, let's say, colloidal science approach, where we also try to use advanced characterization methods to control the structures, size, and efficiency. Just before diving into our topic, this is just some of, these are some of the things we have worked in the past, and sometimes we still work. So I am a physical chemist, so I started working with surfactants and lipid self-assembly systems. And then I jumped into these so-called DNA lipid complexes. So the type of structures of lipid crystalline structures that they form, and also how to come up with new ways of studying their structure and how they let's say behave under flow. So this is also very important for let's say advanced materials fabrication. For instance, coming again to this COVID-19 mRNA vaccines, so they are assembled in microfluidic systems. So maybe further down the road, we would also like to use in situ X-ray scattering to study better how these particles form inside microfluidic channels, and how maybe this can be used to our advantage to tune their structure a little better. So in general, we are physical chemists, and we try to work with systems that are interesting both from a fundamental point of view, but also useful for therapeutic applications. So this is an outline of my talk. So in the first part, I will be talking about so these so-called lipid polymer DNA complexes. So a lot of people call them lipoplexes. And this is because we have a core formed out of DNA or mRNA and a polycation. So there's a polymeric core, and then a complex core, and then there's a lipid membrane put around it, hence this terminology. So we will be talking about our motivation to study these systems, and why we started using this methodology, which will be very relevant, will be a main actor in this talk. So fluorescence cross-correlation spectroscopy. So I will start with an introduction to this methodology, and then how we can use it to understand the phase behavior of these systems. Then in part 2, I will go into a slightly simpler system. And when I say simpler, it's just because whereas here there are 3 components, the DNA, which is negative, a polymer, which is cationic, and a membrane, which is cationic. So here we don't use this polymer anymore. So it's just, it's a system composed only of 2 main components, DNA and cationic membranes. But because the system is simpler, then we can complicate our use of fluorescence cross-correlation spectroscopy. We can embark in a more complex analysis to try to extract more information about it. And then if I still have some time, I will try to talk about a new type of liquid crystal particles, so-called cubosomes. So these are these, let's say, continuous cubic phases when, let's say, dispersed in the form of nanoparticles, and how we can use microfluidics to control better their size. So jumping then into the first topic, so fluorescence cross-correlation spectroscopy has a tool to guide the formulation of lipopolyplexes for gene therapy. And in this work, the key person was Juan Paris, which was a postdoc in our group and is now at the Andalusian Center for Nanomedicine and Biotechnology in Spain. We also have a strong contribution from Ricardo Gaspar and from also Peter De Boyle, which is a colleague of mine, also a senior scientist at INL, an expert in optics. So just a little bit of background, why using this, let's say, complex lipid, sorry, sorry, I don't know what I did here. So sorry, I don't know why my screen stopped sharing. Do you see my screen again?
Yes, we do.
Yes,
okay.
Yes, it's okay.
Sorry about that. Okay, why using these complex designs? Why? And the reason is because in their application, so these particles, if we want to deliver nucleic acids encoding genes into a patient, these particles have to undergo a lot of barriers. So in first place, we need to protect the nucleic acid, so the DNA or mRNA, from degradation, and for that we need a vector. Then we also need to target specific cells. We need them to go inside cells by endocytosis and also get out of the endosomes. And so these are several physiological barriers which are very difficult to overcome with just one simple particle. So we have the example again of the vaccines for COVID. So they work very well for vaccines, but their efficiency, let's say at the single level, is at the single cell level is very low. But they work still very well for vaccines because we just need a couple of cells to be transfected, and then our immune system does the rest. If we would like to try to cure people who have genetic diseases or cancer, we need to bring the efficiency of this transfection a little higher. So because of these multiple barriers, we think that we need something more complex that can perform more functions. And these hybrid nanoparticles seem to be a good solution because we have a polymeric core where we can incorporate different functions like, say, some peptides that once inside the cell help taking the DNA into the nucleus. We can also have some chemical functions that help breaking down the polymer once inside the cell. Whereas at the same time, on the membrane, we can, so in first place, it's more biomimetic, so more biocompatible. And we can also anchor things like targeting peptides or so on. So these more complex designs are good in one way because they provide us with more options for the particles to perform more complex tasks. So this is good. But they are also much more complex to formulate because we have more interactions involved, right? We have now three different components. They are all charged, so there's more pairwise interactions that we have to consider. So our general goal is to understand the assembly behavior of these complex formulations and also bring up new characterization methodologies to understand them. So how are these particles typically assembled? So usually we first start with, by mixing DNA with a polycation to form then this core, the so-called polyplex. And then if we want to, that our liposome to be added to be cationic, so this core has to have an excess negative charge and vice versa. So there has to be charge inversion to drive this assembly to make the lipopolyplexes, right? And I forgot to mention, so the polymer that we are using is polyallysin. This is a very ubiquitous polymer that is not very efficient at transfection, but here we wanted something stable to work with to facilitate the characterization. And the liposomes, they are pegylated, so this means that they have this PEG core around them, and then they also have 80% of the lipid is cationic, 10% is neutral, and then 10% has this PEG on the surface. Okay. So if everything goes well, when we mix these two components, this is what we get, a lipopolyplex. But we must not forget, again, we have to consider all the interactions. We should consider all the interactions involved. So for me, it wouldn't be surprising if in some systems we would get, the liposomes to interact exclusively with the DNA and displace the DNA from the cores and make these lipoplexes. So basically, we would not be forming what we think we are forming. And there's a reason to be cautious about this. For instance, we know that, for instance, heparin, which is a negatively charged polymer, is known to displace liposomes from these complexes. So this is something to be concerned with. And one of the problems is that in most cases, most people working with these systems, they characterize them solely with the zeta potential. So they have this species that is negative, this one is positive, they mix both, and they see if the zeta potential changes from negative to positive. But if we would go from this situation to here, we would also be changing from negative to positive zeta potential. So this is, and the particles also have similar sizes. So this is not a very good way, in our opinion, it's not a very good way of characterizing these systems. So they are difficult to characterize with conventional methods. Of course, you can use cryo-electron microscopy, but that's a difficult tool to use on an everyday basis. And so our question is then, how many particles have all of these three components, and what is the optimal ratio between polymer, DNA, and lipid to obtain this? So for this, we turn then into FCCS, the technique that I mentioned in the beginning. So this is based on fluorescence, colocalization. For this, then we have to label our polymer, our polycation, with a dye. We label it with green. Okay, so our polymer is green. So our polyplex core is green, and we label our cationic liposome with red. Okay, so in this case, takes us red. And so this This technique, FCCS, it's performed on a confocal microscope. And the way it works is the following. So we need some special optics to have the two confocal volumes, so the green and the red, overlapping as much as possible, okay? So these two volumes, these two confocal volumes are overlapping as much as possible, about 70% of overlap volume. And then when we have one particle that is only green diffusing into this confocal volume, we see a spike on the green fluorescence, but not on the red fluorescence detector, okay? So a red particle will show a spike on the red detector, but not on the green. And a particle that has both the red and green, so a particle that, a lipopolyplex that has formed, when passing in this confocal volume, will then show a spike in both channels at the same time. So by looking at the correlation between these two signals, then we can get a feeling of our colocalization. So the percentage of particles that are really of particles that have the two components, to answer our question. So this is the principle of how FCCS works. In practice, this is a bit more challenging. So this is a typical time trace where we are measuring as a function of time these spikes. This is a messy signal, but we see that the two colors seem to be overall correlated. But to have a better quantitative feeling about this, what is typical is to do this procedure, which is called auto and cross correlation. So there's no time to go into all the details, but let's say that this green autocorrelation curve results from the dynamics of only of the green of the species that have the green color, the red autocorrelation curve of the species that have the red color, and here, importantly, this blue curve, it's the cross correlation. So it's the dynamics of the particles that have the two colors at the same time. And from, let's say, the decay time, we can get the size of the particles. But more importantly for us, from these amplitudes here, we can get a feeling of our colocalization. So these are the equations that describe the amplitude of these three functions. They can be complicated if we have multiple components. But just to sum up, we can see that, let's say, the amplitude of the green is inversely proportional. So the amplitudes of the green and the red are inversely proportional to the concentration of green and red species, respectively. And the amplitude of the cross correlation is proportional to the concentration of colocalized species. Okay? So this can be complex, but for the special case of a one-to-one binding between the red and the green species, and if we have no crosstalk between them, these expressions can be they simplify to these simple expressions here. Then by the ratios, so the ratio of the cross correlation to the red autocorrelation, we obtain the fraction of polyplexes that have been coated with liposomes, and the fraction and the ratio of the blue to the green amplitudes gives us the fraction of the liposomes that are being used. So just with a single measurement, then we can get a feeling of then let's say the fraction of polyplexes that have been coated and the fraction of liposomes that are being used. So this answers our question. So these are some of the results. So as physical chemists, we like ternary diagrams. So this is not a phase diagram per se, but it helps us mapping and understand the relative proportions of and behavior of the system. So these axes here are molar fractions of charge, okay? So as we go up in this axis, so this is the DNA polycation axis. So here we have we have excess negative charge, and below this line we have excess positive charge. So this is the line for the polyplexes, and the isoelectric point is at 1.6. And then as we add the cationic liposome, we are going down this direction. And so this triangle here, so we are, the system will always be positive because we are mixing, let's say, a cationic core with a cationic liposome, so will always be positive. In this triangle up here, the system will be overall negative because even though we are adding cationic liposomes, we are not adding enough to fully invert their charge. And here is when we start with negative cores, but we invert their charge to positive. So these are our starting compositions. We have 3 series. We see here there's at the potential. We see that this one here is positive, is at the potential. This one here is very negative because it's further away from the isoelectric point, and this one here is negative, but it has a milder charge. And then we add our liposomes. And this is not FCCS data yet. Okay, this is what is typical is DLS and zeta potential. Then as we add our liposomes, we see that the charge of these 2 inverts changes to positive right away, but we have a region where we see a lot of precipitation. So if we normalize this, we see that this precipitation region seems to occur above this line here. So this is what we call the normalized charge ratio. So if we discount for the part that is neutralized from the DNA, we have still a residual negative charge, and then we do the charge, let's say the ratio of the charge of the liposomes to the residual negative charge here. So we need to here, let's say here, we don't need to add too many liposomes to invert the charge and get a colloidally stable system. For this line here, we have to add a lot of liposomes, a lot of positive charge to get a colloidally stable system. So this tells us about the colloidal stability, but as I mentioned before, it doesn't let us distinguish between the 2 different competing structures. For this, we need to do then the FCCS. Again, so our polycation is labeled green, and our liposomes that are also cationic are labeled red. So this labeling scheme prevents distinguishing from the possibility of forming lipoplexes, because the two dyes, the 2 labels are in species of the same charge. So we only see colocalization between them if we really form the particle that has the 3 components, because DNA would have to be in between to associate these 2. And these are the results. Then these are the ratios, the ratio of the cross correlation to the red autocorrelation, which, as mentioned before, gives us the fraction of coated cores, and the ratio of the cross correlation to the green autocorrelation, which gives us the fraction of fused liposomes. And we see that when we start here with a positive core, we have a very low fraction of each. So basically the association is very poor. There's nothing really being formed. But when we go to this system over here, so when we start with a mild negative charge, we see that now we have an excellent fraction of polyplexes that are being coated by a liposome. We see that the fraction is actually starts at 1, and it actually goes a bit above 1 to about 1. And this is because in theory should only be 100%. 100% should be the maximum, but this is because our simplified model are assuming 1-to-1 stoichiometry between both. So in practice, what this tells us is that when we add a lot of liposomes, probably we get some cores that have more than 1 liposome around them. And at the same time, we see that as we keep adding more and more liposomes, they will just be in excess. So the fraction of liposomes that are being used is just decreasing. So this seems to make a lot of sense. And these two samples because when they are not colloidally stable, they cannot be analyzed with FCCS. We see the same thing. We see that the colocalization is quite high, fraction of coated polyplexes is quite high, and we also see that we are using a lot of liposomes, more than we would have needed. So a lot of liposomes are in solution. But we have to add all of these. Remember that before the system is unstable. So this probably means that in this system here, there's a lot of free DNA, and we need to neutralize that free DNA to get a stable system. Once we understood this, then we could be confident to look at our, let's say, our electron microscopy. So I don't like to use electron microscopy as the only probe, because we cannot probe all the solution, let's say, all the volume of the sample. But once we are confident about this, we can also be more confident about electron microscopy, and it seems to corroborate well the observed results for FCCS. Here we have a free polyplex, here free liposomes, and then in the cases of lipopolyplexes, we seem to see these membranes that are more flexible, and in some cases with this sort of core inside. These are dried samples, that's why it's a little trickier to analyze. Okay, so FCCS confirms that these lipopolyplexes are forming, and for them to be formed, we require a charge inversion. So practically all the polyplexes are converted to lipopolyplexes, and this helps us then getting these guidelines for optimal assembly. So now we know that we have to be in this region. So we confirmed our hypothesis, but can we understand the system better with FCCS? So since we see a very small deviation from one-to-one stoichiometry, if we do the ratio of the two individual autocorrelation curves, we also get the ratio of the total number of liposomes to the total number of polyplexes. And if we plot this here, we see that, so I forgot to mention, so this shaded bar is regions where the solutions are stable some of the times, and some of the times are not stable. So this is let's say marginal stability. And we see that when we plot then the ratio of liposomes to polyplexes, we get this extrapolation that we reach colloidal stability when we have this one-to-one stoichiometry between the number of liposomes and the number of polyplexes. Okay. This is different from a charge stoichiometry, right? So the charge stoichiometry we know very well because we calculate the number of lipid molecules, we weight them, and we know the composition of our systems. But knowing the stoichiometry between liposomes, which they can be somewhat polydispersed and so on, this is a lot trickier. And you know, in the beginning, we were looking at this normalized ratio, and we were saying, oh, we need a lot of excess positive charge to stabilize these systems. But it turns out that if the liposomes, if an individual liposome has enough charge to neutralize one individual polyplex, then this is the critical number that we need. It's a one-to-one ratio. And this is very difficult to estimate just by calculations. We really need to measure because it depends on the size of our liposomes. So just to conclude this first part, so I need to speed up a little bit, but this was the longest, I think. So FCCS is a powerful tool for complex characterization. And so besides allowing us to obtain quantitative information about this core shell association, which is difficult to obtain with traditional techniques, it also gives us additional insights into the forces and factors influencing this association. So we learned that electrostatic forces are indeed crucial. We really need to have charge inversion for this process to occur, and we need at least a one-to-one ratio between the number of liposomes and cores to avoid precipitation from instabilities. Let's go to the other system now. So, like I said, these are apparently simpler colloidal systems because we only have cationic liposomes and negative DNA. They are still promising, they're still relevant. So the COVID vaccines are somewhat similar to these systems, but there's still a lot of room for improvement. And one of the reasons also is that there's a lot of experimental questions that are still difficult to answer with, let's say, the more common analytic methods. For instance, one question we were wondering about was how many DNA molecules can we have per nanoparticle? This is difficult to answer experimentally. Usually we only know the encapsulation efficiency, but the number of DNA molecules per nanoparticle is trickier. But this is important for pharmaceutical applications because it influences the potency, and also if for an application you need more than one type of DNA or mRNA, and you need to co-deliver, then it's a parameter that you need to know to optimize. So as I said, the mainstream analytic methods are not very useful to answer this question. For instance, the NLS gives us only the size of the particles. X-ray scattering gives us, let's say, average distances between DNA molecules, but it's tricky to answer exactly how many molecules we have. And cryo-TM gives us a very good view of the structure, but we cannot count individual DNA molecules inside of this. Again, we use FCCS. Now our DNA is labeled with green, and our liposomes are labeled with red, and then we make these, let's say, onions that have both red liposomes and green DNA. And in this case, we cannot use this simplification of one-to-one stoichiometry because they undergo a complex M to N assembly where M is the number of liposomes and N is the number of DNA molecules. And we should still be aware that we can have also in solution free DNA and free liposomes. So the equations become, in this case, really tricky to answer. Okay, so this stands for free DNA, N is the number of DNA molecules. This Q is a factor that has to do with possible variations in intensity when the DNA is condensed and so on. And this is the concentration of the Lipoplexes. And it's the same for the other equations, okay? So here we can use this knowledge from other methods that when we have a good amount of excess cationic liposomes, and this is important because in therapeutic applications, we tend to have an excess of cationic liposomes. There is virtually no free DNA in solution. So when this is the case, then this equation here becomes much more simple, becomes simpler, and just from the amplitude of the green autocorrelation, we can get the number of these lipoplexes, okay? So once we get this, we can rearrange this equation to also this useful expression that gives us this quantity just just using this relation between all the amplitudes, okay? And once we get also this quantity, we can rearrange this equation. So now we know everything to extract the number of free liposomes. Since we know how many liposomes we started with, we can calculate the percentage of used liposomes. And we can also estimate the number of liposomes used per particle. And since we also know how many DNA molecules we started with, we can also now by knowing this number, we can then estimate the average number of DNA molecules per particle. So to recap, just by knowing these three amplitudes, we can know the number of lipoplexes and free liposomes. And by knowing the initial compositions, we can extract the average number of liposomes and DNA per particle. So we varied the membrane charge density of the liposomes. We also varied the ratio between liposomes to DNA. And sorry, I'm speeding up a little bit now. And we get this very interesting graphic that shows us then the average number of DNA plasmids and cationic liposomes per particle. And what is interesting here to see is that so this is the charge ratio. In this regime, we have excess DNA. And when we have excess DNA, we see that we have a larger number of DNA molecules when we have membranes that have a smaller cationic charge density, okay, than membranes that have a high cationic charge density. And this is because since we are overcharging, we have too much DNA, having then membranes where the cationic charge is more dilute, makes it easier for DNA molecules to pack into these membranes because there's more space between DNA, so we can have a higher overcharging. But when we go to excess cationic charge, now we have extra liposomes. Now we see the opposite happens. So now we see a higher number of DNA molecules when our DNA, or when our membranes are more densely charged, have a higher cationic charge density. And this is because now we have a smaller number of liposomes. So again, the cationic charge is fixed. But here we have a smaller number of cationic liposomes, and therefore we are distributing the same DNA by a smaller number of liposomes, and therefore we have a higher number of DNA here. Regarding the number of liposomes used, I would just like to highlight that this is very similar to previous data that we had, where we estimated the number of membranes with X-ray scattering. So the absolute numbers are higher in the case of X-ray scattering, and this is because here we had to use more concentrated samples, but the relative trends are the same. In particular, here is where we have the smallest number of membranes. It's also here for the same conditions. So this is important because if we want to optimize, for instance, for having a higher number of DNA plasmids, this approach really tells us what are the conditions that we need to have. So we can also measure the size of the nanoparticles through the shape of these curves. Through this, we can get the diffusion coefficient. And here we compare these sizes with DLS, and we see that we have a consistent trend where the sizes determined with FCCS are smaller than with DLS. And this is because of polydispersity. So DLS is very sensitive to polydispersity. FCCS also, but in FCCS, since we have a very small confocal volume, which is 0.25 femtoliters, we are in a regime where we are looking basically at just one particle at a time. And therefore, let's say we are almost effectively looking at the number average compared to the DLS. So to sum up, so again, FCCS is quite useful. And it's really a tool that lets us get extra information about the relative amounts of our system. And as a side, let's say as a bonus, it also lets us get the size of our particles. So maybe I can still talk very quickly about microfluidics, or is it too late?
No, it's okay. Go on.
Okay, I'll try to make this quick. So this data, this is data also. So this is a project that has been run by students under my supervision, of course. But here we wanted to use Cubosomes and microfluidics to control their size. And part of the motivation for this is that microfluidics are now used to better control dissociation in the fabrication of lipid DNA nanoparticles. So eventually you also want to use lipid DNA in our systems. And in this work from Peter Collis, you can see that mixing the lipids with DNA under these conditions, you can come to conditions where you have a minimal polydispersity, so very reproducible particle sizes. And importantly, we also got inspired by this work from these two labs at NIST, where they via controlled mixing, they could tune the sizes of the liposomes. So basically, when the mixing is faster, they can have liposomes that are increasingly smaller. So we wanted to do this for cubosomes. So cubosomes are these very interesting versions of bicontinuous cubic phases. And the way they are typically done, so this is the phase diagram. So here we have the cubic phase coexisting with excess water. And a stabilizer like F127 polymer is added. So this is an amphiphilic polymer. And we add ultrasound. So basically we keep breaking the cubic phase into smaller and smaller particles in the presence of the stabilizer, and the stabilizer goes around the particles and stabilizes them against aggregation. So this method is not so efficient to encapsulate some molecules that are sensitive, and the particles are also not very monodisperse. An alternative is to do this using solvent exchange. So essentially, the lipid that forms these systems is previously dissolved in ethanol, and then the ethanol with the lipids is injected in a solution. And so the ethanol is a good solvent for the lipid, but water is not. And as the ethanol mixes with water and becomes in minority, let's say the self-assembly of these lipids occurs. And if we have then this stabilizer in solution, it will tend to stabilize the particles at a certain size. Also here, we don't have a very good control in the size. So we wanted to do this in microfluidics. So we put our monoline in, or phytentry also, different lipids in ethanol, so about 1%, and then we mix them with water carrying this stabilizer. And because we have, let's say, this sort of confinement, the conditions are such that the flow is laminar, and the mixing occurs in a well-controlled way, where the mixing between ethanol and water occurs at this interface here. And what we see is that then, as we increase the flow rate ratio, so as we increase the ratio of water against ethanol, and we make this stream increasingly narrower, we decrease the size of the cubosomes. We see that we can go from a situation where we have about 220 nanometers to 150 or so. This is just this is an extremely simplified model, okay? I'm not talking to physicists, I should be careful. But we just wanted to see if our hypothesis was okay. We need to improve this, of course. But we just postulated that, usually in these situations, the size is usually in nucleation, right? These particles are nucleating from solution. The size is usually proportional to the mixing time. So faster mixing, usually you get more nucleation spots and smaller sizes. So, and our mixing time is proportional to the square of this this narrowed stream width divided by the diffusion coefficient. Okay, because this is the distance that the molecules have to travel to achieve mixing, and this width is proportional to the width of the channel divided by the flow ratio, the flow rate ratio plus 1. And if we put all of this together, then we see that we sort of get the right trend to explain this size variation. And also importantly, So if we have some initial water in this initial stream, so we have a little bit of water here, so that let's say the time needed to obtain let's say the water concentration to induce assembly, the time is shorter. So we also see a shift, a consistent shift in all the sides. We would like to improve this model, basically with nucleation theory. We haven't had the time to do that yet, but I'm quite pleased with the ability of tuning the sizes of these particles just by adjusting, essentially just by adjusting the flow rate, the flow conditions. So just to conclude this part, microfluidics provides a feasible way of producing monoline nanoparticles of controlled size, and by manipulating the flow conditions and the composition here, I mean by adding slight amounts of water in the initial ethanolic solution, we can tune the size between 100 and 220 nanometers. And this is a very relevant size range for therapeutic applications. So this is interesting. Okay? So we need now to improve let's say this theoretical model for sure, but we are also currently working on improving the scalability of this method. So having a higher amount of Cubosomes produced. So this is all. I have to thank, of course, the people that have done the work, notably so Juan and Ricardo in the FCCS work, in the microfluidics, Celso, and some other master students. Also previous mentors and, of course, funding, which is always important, and both provided by the FCT and INL. And I also thank you for your attention. Thank you for listening. I'm happy to take questions.
Okay, thanks very much, Bruno, for a very informative seminar. And it's now up for questions. So who would like to ask a question, make a comment, voice praise, criticism, present suggestions, whatever? Just raise the little yellow hand and ask your question. So who wants to be first? Come on, guys, this is your chance.
There was maybe something not so clear. I'm happy to clarify also.
So who wants to ask a question? Well, maybe I can start. Well, this is a very general and rather naive question. But right at the start of your talk, you mentioned making these particles of all sorts of different shapes. And I'm just wondering, how do you know exactly what shape your particles will be? Does this have to do with the shapes of the building blocks themselves, with the way you make them?
So this was right at the beginning.
Well, yeah, I think it applies generally to other parts of the talk. But right at the beginning, you mentioned these particles, and some were just capsules, some were onion shapes, and I'm just wondering how you know which shape you're going to get. Is it from the shapes of the building blocks?
Yeah. Let me just fetch that slide. Just a second. Sorry, I'm having a bit of trouble with this sharing now. Share screen. I'll just share the screen. You mean this slide, or?
Yeah, I mean things like that, or when you mentioned at some point I remember some particles which were, well, onion shaped, where you had alternating layers of different materials.
Yes. And
the question is, how do you know they're going to go into precisely those shapes and not something else?
Yes. Let's say the internal structure here, for this internal nanostructure is for the most part controlled by the shape of the lipid.
Okay. Okay.
So the shape of the lipid, so the intrinsic curvature of these membranes tends to control the type of internal nanostructure, much more than DNA. So these three structures they form. So when we have lipids that have a cylindrical shape, which is the most common situation, we have these bilayers. And then if we change this curvature, this is simply by controlling the ingredients that we have of the lipid.
Yeah.
Okay. And then let's say at a more meso level, so when we zoom out a little bit to come to these structures here, this we don't control so well with self-assembly, and this is one of the reasons why we want to use microfluidics to try to control this better. So this is controlled by a lot of factors. Of course, it's controlled also by the, let's say here in this diagram here, just by the ratios that we have. We can go from a situation where we have massive precipitation, which is in this region here, to a situation where we have particles that are colloidally stable. But then, let's say if this is a perfect onion, or if it has defects that we are not being able to control yet, that we can only see, that type of information we can only see with electron microscopy, in a way of speaking. But it's something that is important to control also, and this really sets one of the motivations to use the microfluidics, as we were using here, is to control. So here we don't control the internal structure, right, of these cubosomes. The internal structure is set by the type of lipid that we are using.
Yeah.
But by controlling the flow conditions, we can obtain the different sizes. But it's not a, let's say, it's a significantly empiric world when we try to control the mesostructure.
Okay.
There's a lot of experimenting around.
Yeah, but now coming back to the end of your talk, where you mentioned using mesophases. Now, if you have well characterized mesophases, then in principle that would give you a better idea of what shapes you should be able to get. Is this right? Then in principle, at least you could have a little more control, and in principle, I think you would need to do a little less trial and error. Is this correct?
Yes. Sorry, maybe I didn't understand initially your question. So let's say the shape of these particles will be cubic, as we see here in this, right? But I meant the size that we don't control. Here, going to a different example now with these nanoparticles, we see that they tend to be spherical, but there's these defects here. So again,
yeah, can you share your screen, please? Because I don't see your presentation.
Oh, sorry. I will just keep my screen shared. So you see my screen?
Yeah, I do now.
Okay. So from the lipids, we know that we should expect bilayers.
Okay.
And if we have bilayers, we should expect phases, which, let's say, at the internal structure level are these, stacks of lipid bilayers with DNA sandwiched in between. At the mesoscopic level, the fact that these are layered also tends to form this sort of onions, right? But we see that the structure is not perfect. There's these defects here. So this has to do with the way the particles are forming. And this part is difficult to control completely, you know, to have something that is completely defect free, or controlling the total size of this onion, you know, if it has really, you know, if it can be easily, let's say, 50 nanometers larger or 10 nanometers smaller. That part is very difficult, but the overall morphology, you know, being layered or being cubic or being hexagonal, that depends on the ingredients that we have decided to work with.
Okay.
I don't know if this was this answers the question you asked.
Yes, it basically does. Yes, thank you. Right. Okay. So, any more questions, comments, praise, criticism, suggestion?
I can just comment a little bit. So I'm commenting my own things. So maybe this is the right place to. So when we deduced these equations, we assumed just, you know, no polydispersity. I maybe, you know, someone with a better physics background, if could incorporate polydispersity here, let's say like a region or of, you know, where we don't have just an average number of membranes and DNA, these model predictions could become a lot better. Just an observation of our model here. So this was a limitation in our model which we didn't know how to improve, for instance.
Okay, so what I think you're saying is that, in your opinion, there would be scope for collaboration with people of, say, a more quantitative persuasion.
Yes, for sure.
Okay, very good. Right, so let me ask one last time. Are there any other questions or comments? Well, apparently not. But we'll probably be getting back to you sometime in the future.
Yes, since you will have the video with you, if you want to review, and, you know, and maybe I talked a little quick, so please.
Oh,
please
just yeah, it's just a lot of information. It's just a lot of information.
Yes.
Right, so I think it's now time to wrap up. Okay, thanks very much, Bruno.
Thank you again for the invitation and for your interest.
Our pleasure, and we look forward to meeting you again in the future, hopefully in person. Let's hope you'll be able to visit us in person at some point.
Yes, for sure, that would have been something I would have liked, but, you know, this part of the year now became very complicated. And
yeah, sure, no problem. Okay. Right, so that's it. Thank you all for coming, and this was the last seminar of the current semester. We shall now break for Christmas and New Year, and also for exams. So we'll be coming back sometime in late February with more seminars for you. So happy Christmas, happy New Year, everyone, and we'll see you then.
Yes, happy Christmas and happy New Year to everyone too.
So recording stops now.